Yep. Okay. Leave the floor to you.
Do you mind? I might just rotate it the other way.
Yeah.
Hello. I'm super happy today to be in front of all of you guys. It's very cool to be around a bunch of Cyano people. You've all, from what I've heard, have been doing very impressive stuff. So very happy to be here. But as impressive scientists as we all are in this room, we're never going to beat biology itself when it comes to engineering. Biology has time on its side. Over generations, evolution can make pathways and regulatory systems that we have to spend years just to comprehend. So we're never going to beat biology in that way. But there is one caveat. Biology is better at us than engineering only for organisms that have evolved in the environments that they're in. For example, I'd like to talk about Synechococcus PCC 7002. This strain was isolated from mud at the bottom of fish pens. And these fish pens, like a lot of the fluctuating environments that many cyano are isolated from, experience a lot of seasonal fluctuations in temperature, light, and nutrient availability. So this is measurements of nitrogen fluctuations in one fish pen in particular, and you can see that the amount of available nitrogen peaks in the fall and drops drastically all of the rest of the time. So essentially, these cyano have evolved to deal with very high peaks of nitrogen, but then long periods of months at a time without nitrogen. Of course, they've evolved here, so they are very well equipped to endure these conditions. However, if we want to take these cyano, which are doing great in mud pens, and put them somewhere else, like a high-rate algal pond, like one of these, the cyano aren't as well suited for this. These ponds experience much more rapid fluctuations. They're great for cyano if you want to use the cyano as bioproducers or even a method of treating wastewater, but instead of nitrogen fluctuating on the scale of multiple months, it fluctuates on the scale of a couple days. So this is a very different environment from the one that cyano has naturally evolved in, and so therefore the cyano aren't as well suited to these conditions, which we might want to use them for bioproduction. So if we're looking to fix this, we could wait a bunch of generations for evolution to do its work, or in the meantime, we could try and make a more specialized fitness for a strain of our own. We could do this, make a strain that could survive in these more rapidly fluctuating environments by identifying genes that might burden the cell in typically fluctuating environments, but could actually be a benefit in rapidly fluctuating environments. So essentially, if you can find a mutant that maybe it doesn't have some of the genes that code for the robustness required to survive months of nitrogen deprivation, but maybe you don't need that when nitrogen is only ever deprived for a few days at a time. This could help us make a strain that is specifically designed to do very well in bioproduction. Now, when we talk about doing very well in bioproduction, we should talk about what that means. Essentially, you would want to maximize output while minimizing input. So maximizing output would be ensuring that the cells grow faster and bigger, and minimizing input would be making sure that the cost and energy requirement to maintain these cells is as low as possible. So you'd want to not have to change the temperature too much, not have to change the light too much, and ideally they could thrive off as few nutrients as possible. Specifically for my talk, I'm going to be talking about how we can maximize growth rate while minimizing nitrogen input. So to talk about minimizing nitrogen input, I should first give you guys a rundown of PCC 7002's nitrogen deprivation response. Adrian did most of the hard work explaining this, but I can run you guys through it again. The nitrogen deprivation response is heavily dependent on 2-oxyglutarate, which is created during carbon fixation and consumed during nitrogen assimilation, which makes it a pretty good indicator of the carbon-nitrogen ratio for a cell. Now, when cells are lacking nitrogen, 2-oxyglutarate builds up because it's no longer being consumed through nitrogen assimilation. Does anyone know what that was? Okay. When 2-oxyglutarate builds up, it's able to bind with NTCA, which sends the cell down a lot of its nitrogen regulatory pathways. However, for NTCA to send the cell down those pathways, it has to interact with PIP-X, and it can't because PIP-X is bound by P2 when 2-oxyglutarate is low. But as 2-oxyglutarate builds up, it breaks the connection between P2 and PIP-X, and it helps NTCA bind with PIP-X. From there, NTCA leads to the buildup of glycogen and the buildup of catabolites used to break glycogen down. This is able to facilitate that response that Adrian talked about, where the cells store glycogen while they're unhealthy, they build it up, and then as soon as there's nitrogen, they can go back to, they can basically get back to healthy very fast and recover fully. NTCA also interacts with NBLA, which initiates phycobiliprotein degradation. Breaking down these blue-green proteins sends the culture from this nice healthy green to a slightly more sickly yellow. Now, as Adrian talked about, there are lots of mutants that lack this response. Specifically, if you knock out some of the key players in the nitrogen deprivation pathway, like PIP-X or NTCA, you can see that the cells bleach a lot less. Similarly, if you knock out glycogen storage, you're not going to see bleaching. And these two mutants are knockouts of something connecting glycogen to the central carbon metabolism, as well as something used to synthesize to synthesize glycogen. And without these genes, you can see that the bleaching is completely gone. However, because this bleaching is a beneficial response used for survival, almost all of the mutants cannot survive nitrogen depletion as a result, even when nitrogen is resupplied. However, you'll remember Adrian also talked about a carboxysomal mutant that doesn't bleach. This one's really interesting because it's slightly less inherently tied to that nitrogen response. It's still connected through carbon fixation, but it's a little less hooked in, and you can tell that the delta CCM, grown, of course, in high carbon dioxide, because that's the only way it will grow, doesn't bleach even when the wild type does. So I wanted to see if this mutant, which is lacking the chlorosis response that is essential to cell recovery, can recover without this chlorosis. So what I did was I grew up the cells through log phase and into stationary phase until they were overgrown and starved themselves of nitrogen. Then at that point, I diluted them down with fresh media and nitrates to see if they could regrow after dilution, essentially recovering from nitrogen deprivation. And you can see here on this plot of optical density that they definitely do. You can see them first growing through log phase, entering stationary phase at an optical density of about 0.5, and then after dilution, they recover with honestly minimal lag. So the question is, if the wild type is breaking down all of these pigments just to survive, but other cells can survive without doing that, is chlorosis even an advantageous response to nitrogen deprivation? To explore this, I basically repeated the same experiment, but with the wild type as well. So I could compare the growth rates between the wild type and the delta CCM at after nitrogen resupply. So you can see here, this time point 0 here is immediately after dilution. So this is after the cells have been deprived of nitrogen and then they're just starting to regrow. And you can see the delta CCM overtakes the wild type very quickly, which is quite surprising because we wouldn't even expect them to be able to survive, let alone thrive. The question is, these have occurred in separate cultures. We have the delta CCM in one flask and the wild type in the other. I was curious if this would confer a fitness advantage in co-culture. And looking at co-culture specifically when you're looking for a fitness advantage is important because you're basically making sure that all of the confounding variables that could exist between the two strains are exactly the same. They have the same light availability in the culture, and perhaps more importantly, they're competing over the same exact resources. So to run this co-culture experiment, I took the wild type and delta CCM and inoculated the same flask with both of them. I let them overgrow into nitrogen deprivation, and then we would assume at this point that the delta CCM probably isn't bleached while the wild type is starting to bleach. So then when nitrogen is resupplied in the form of fresh media, the delta CCM is going to double much faster than the wild type, as suggested by those growth rates I just showed you. From there, I can measure the allele frequency of the wild type and the delta CCM using microscopy. The reason that I can do this is that these strains are GFP tagged. Their Rubisco has GFP. So the wild type, which has its Rubisco concentrated in these carboxysomes, can be identified by clear, bright, concentrated yellow marks, while the delta CCM, which has the Rubisco diffuse throughout, just kind of has a faint yellow glow throughout. So using this information, I can take a picture like this and identify how many wild type and delta CCM cells there are in each picture. From here, you can calculate the allele frequency pretty easily. So I was curious how the allele frequency would shift if I ran through multiple of these fluctuations. So instead of growing them up, depriving them, measuring the allele frequency and calling it a day, I went through multiple of these. I first grew them, and then at the end of log phase here at time 0 or at point 0, I took a microscopy measurement. Then I allowed them to oversaturate so they were out of nitrogen, diluted them down, and started regrowing them. And at the end of their growth, took another picture, and I repeated that a couple times. And you can see at each of these points, I measured the allele frequency of the wild type, and you can see the delta CCM has a very clear fitness advantage. It starts at about 50%, which is what I inoculated it at, and practically halves with each following cycle of nitrogen deprivation and resupply until after just 3 cycles, it's down to about 10% of the population. So this mutant, which can't even grow in low carbon dioxide, at high carbon dioxide, has a huge advantage in these rapidly fluctuating environments. Now, this is a mutant that is, again, only tangentially connected to the nitrogen response. So it makes me curious how many other mutants could also give an advantage to a strain, how many other mutants could give an advantage to a strain if they were applied to the same genome, essentially. So there are lots of other mutants that could be used to tune PCC7002's resilience to rapid fluctuations, whether that's for bioproduction or any other purpose. And now the trouble is just finding them. Now, the approach that I used to validate the difference between the delta CCM and the wild type is not one that I would want to use to identify every mutant, and there are a few reasons for that. First of all, the framework of nitrogen deprivation through overgrowth and then nitrogen deprivation is just clunky. We know that nitrogen is what is guiding this chlorosis, and so there's no reason not to just put the cells into nitrogen deplete media and then go from there. This gets rid of waiting for them to oversaturate and dilutions. But second of all, this framework that I explained before only really allows me to investigate one mutant at a time. And it's dependent on GFP tagging, which not only can skew results of the fitness advantage difference, but it might not even be possible for every mutant. And as someone who spent many, many hours categorizing and counting cells, I can say that this is very time intensive. So going forward for something more high throughput, I'd like to use a different strategy. And I think a pooled CRISPR screen could be a really good way to do this. Instead of taking one mutant at a time, we could take a whole collection of mutants, put them straight into nitrogen deprivation. And then at this point, any cells with this specialized fitness that we're looking for will probably be doing better. And so when we add nitrogen back in, those cells will double faster than the rest. Then instead of sampling with microscopy, I can sample for sequencing. And this way, not only are we able to investigate multiple single mutants with one experiment, but we also don't have to depend on GFP tagging, which can be tricky. Now, I think this experiment would have a lot of potential for revealing some really interesting things, both about the function of a lot of these genes and also guiding how a more fit strain could be made for rapidly fluctuating environments. But for something this high throughput, we would want very fine-tuned initial screen conditions. So what I've been working on for a lot of this summer is exploring different conditions that might impact the fitness difference between the wild type and the delta CCM to try and find basically the most informative conditions, whether that's maximizing the difference or just identifying places where something interesting happens. So by changing light, temperature, deprivation time, or carbon availability, we can see how the dynamics between the wild type and the delta CCM change. For this summer so far, I've been prioritizing light and deprivation time, both because they're simple to change, because I think temperature isn't going to be as informative, and with a specifically high CO2 dependent mutant, varying carbon availability is going to be slightly more complicated. And I've gotten some pretty interesting results so far. Looking at different light levels, these are somewhat preliminary results. I only looked at 2 of them so far, but essentially the framework for this experiment and the framework for every experiment, but just with the conditions tweaked slightly, is I take the wild type and the delta CCM, I put them in separate nitrogen deplete environments, and for a set amount of time based on how long I'm trying to deprive them of nitrogen. And then at the end of that period, I add nitrogen back in and I plot their growth over time. So these dots right here are the optical density measured at however many hours since nitrogen resupply. And these lines are the exponential fits of the growth at each light level. So you can see even just visually that the wild type grows much slower than the delta CCM, more so at 60 microeinsteins than at 130 microeinsteins. To look at this even more quantitatively, I approximated the growth rate by taking the coefficient of the exponent, and you can see those plotted here. So we can see that, interestingly enough, the delta CCM's growth rate stays mostly consistent between the 2 light levels, but the wild types increases at a higher light level, mitigating the fitness difference. I'm not entirely sure what's going on here. I think it's very possible that the delta CCM is not limited by light, but by carbon dioxide. And so it's possible that it's already functioning at capacity while the wild type is limited by light. And so increasing that light helps it recover faster even from the bleaching. So that could inform different light levels going forward. I definitely want to get a slightly bigger range, both below 60 and above 130 to see the specific dynamics of this trend. And then I also looked at nitrogen deprivation time. Because I ran experiments at 0, 10, 24, 48, and 72 hours, I'm not going to show you all of those exponential regression plots, but I got these growth rates the same way. And once again, you can see that the delta CCM's growth rate is largely consistent across each time point in pink here, while the wild type's growth rate decreases drastically the longer you leave it in nitrogen deprivation, to the point where at 72 hours, in the 48-hour period I looked at, the wild type hadn't recovered at all. So there was essentially no growth. I'm really curious about this. I do wonder if presumably there has to be some point of nitrogen deprivation that the delta CCM can't recover from. It just doesn't necessarily show signs of slowing down here. And you can also see I plotted the ratio of the growth rates between the delta CCM and the wild type. The higher the ratio, essentially, the higher the advantage for the delta CCM. And you can see that the advantage increases drastically with time. So I would be curious to do points even farther along this line. So I have a lot more that I'd like to do. So far, I've looked at 2 light levels across 1 nitrogen deprivation length and 5 nitrogen deprivation lengths across 1 light level, but I'd love to fill in the rest of this matrix and find more perhaps interesting points to look at. Maybe it would be beneficial to both maximize and minimize fitness difference. I'd be interested to see if light or deprivation time has more of an impact. And much further on down the line, it'd be interesting to see how these dynamics vary across gas concentrations, as well as eventually moving back into co-culture and seeing how number of fluctuation cycles impacts things. And I think that with these very diligently found conditions, I can select a couple that would be informative potentially for a CRISPR screen, and a finely tuned high throughput CRISPR screen would help us not only uncover the functions of a lot of genes, but also tune a specially fit nitrogen stress resilient strain that could be very helpful for both bioproduction and a bunch of other applications. So I'm really excited for this, and I'd love to answer any questions you guys have.
So much.
Mhm.
I can start.
Mhm.
I think you have a really interesting project like, yeah, and one question I have is that what's your hypothesis on the 600 microinch condition? Do you think very high light or, yeah, relatively high light will like decrease the beneficial of this delta CCM?
Yeah, I definitely think so. That seems to be the way the trend is going, especially just anecdotally. I've looked at the delta CCM grown in -N in highlight, and it does bleach faster, which makes me think that it would also be limited in recovery, whereas the delta CCM might be able to recover, although I think at like that high, it's not advantageous for either cell in general. So I definitely expect the delta CCM to do worse. I don't know if I'd expect the wild type to do better. Yeah.
Yeah, because I think the carbon and nitrogen balance is one big like balancing thing in the cell, and also the light and the CO2. Also, because when you delete the shell, then the CO2 like the CO2 supply is kind of low in the cell, and then you don't need that much light. And even if you use medium light for white type, it can generate kind of ROS like in the cell for your delta CCM mutants already. So yeah, when we talk about highlight and low light, it's really like dependent on how fast the cells can take CO2. Yep.
Just to just a quick comment on this point.
Yeah.
The very first thing we did 3 years ago when we discovered this phenomena
Yeah.
was using a multi-cultivator from PSI. I don't know if you know. Yeah, you know tools. Anyways, we were following the OD in response to different treatment of deprivation, and we were using, I believe, 500 microeinstein, which is twice higher than saturation for these strains.
How
And
much? I didn't get.
I think you said 500
Okay.
microeinsteins.
500. And the mutant still bleached. I mean, the mutants and the wild type bleached faster, but the mutant bleached slower than the wild type. And in this context, this is the first context where we saw an advantage of the mutant over the wild type. So I'm not sure how I think in terms of this 130 microeinstein might just be, as Julia said, that we are working here in conditions where the CO2 is limiting for the regrowth. But in context where I'm 100% sure that CO2 is not limited, even at highlight, I don't think the ROS production in the delta CCM mutant is any problem for them to regrow afterwards. And they still regrow faster than the wild type.
Okay. Okay. Yes.
I didn't really follow your screening part. So when you have the multiple mutants, we mix them together, and then so what do you screen for? Is it just how they growth or survival or?
Yeah, it would be survival. That's the one tricky thing about this is the degradation of the phycobilid proteins is very informative. It's not the only thing informative because, like I talked about, there are definitely cells that don't bleach but also don't survive. But it's still something, and we'd miss that non-bleaching phenotype entirely with a pooled screen because they'd all be mixed together. But I'm looking at this in the context of cells that can grow faster in rapidly fluctuating nitrogen conditions, because that's what would be relevant for bioproduction. And so if that's the case, I think step one would be just looking for fastest growth. Afterwards, you'd maybe want to look at like cell size and things like that. But I think survival and outcompeting other strains is a good indicator of fitness.
So you would take the end product that is go for sequencing at different time points?
Yeah, that would be the idea. Mm-hmm.
Thank you.
Maybe it's very stupid question, but in this case, you're still for growth, as you say, but
Mhm.
in the future, you're gonna move for cell sites or other features. How you would redesign your experiment to do so?
It's a really good question. It would, to my knowledge, definitely need to not be pooled. I think a way to make things more manageable, it would be if I proceeded with this first, you can identify genes that might be helpful because like you're not going to want any mutants that do far worse than the wild type. So I could do a screen to eliminate a bunch of genes or a bunch of mutants that basically aren't relevant. And then from there, it would maybe be a little bit easier to individually grow these in nitrogen deplete media, resupply, and then look at cell size or any other factors that I'm interested in.
And also potentially a stupid question, but if you're
Not at all.
trying to make a strain that is for bioproduction, do you think it's worth focusing on strains that perform better in the conditions we know we're going to want to use for bioproduction? Which, yeah, I don't know. It's very interesting going into everything else, but broadly, we know what we want it to do, so why don't we try and make it perform the best in that condition?
Yeah, that's a very good point. It's definitely something I've been thinking about. I don't know. Presenting is always nice because it makes you make sure that your logic is right. And that is something I've thought about is instead of like I talked about it a little bit here, but then I didn't necessarily follow it. Where you're going to want, you know, growth that is as cheap as possible, basically. And I need to do more research into specifically what that looks like, but you're right that we'd probably want to optimize things at those conditions. It's also possible, I don't know, if we make some super strain that grows way, way better at slightly different conditions, but it's still cheaper overall because of the lack of nitrogen, then that's worth doing. But I should look into typical conditions for bioproduction so that I can keep that more in mind. Thank you.
If not, then thank you again.
Thank you guys so much. I will continue with my own. Yeah, yeah.
So what are your ambitions? You want to live and growing minimal density, right? But at the same time, you want to grow enough biomass. So what are your constraints? You then, no matter what you do, aren't you just limited by the biomass, it's a nutrient content? Are you aiming to make mutants that, what can I say? How can they use less nitrogen? That's my question. Are you aiming to make mutants that make less proteins, make less pigments, because something should be missing, right?
Mhm.
What are you expecting?
Yeah, I think while it might be more challenging to make something that gets by with less nitrogen in general, it might be more helpful to model these rapidly fluctuating conditions instead. I think better fitness in rapidly fluctuating conditions more models what you would expect from a bioreactor or something designed for bioproduction as compared to these more natural environments. So I think it might not be about limiting nutrients entirely. I'd have to quantify if that's something that you could do. I don't know if like periods of nitrogen depletion would actually be beneficial, or sorry, I don't know if periods of nitrogen depletion specifically to save money on nitrogen would necessarily be beneficial for biomass in general. But I do think that having cells more specifically designed for rapid fluctuations prepares them much better for bioreactors. And I also think from a more like pure science standpoint, I'm very interested in just having the capability to tune cells for very different deprivation times. I think that's very interesting and could have a lot of applications that have kind of yet to be seen.
Can I comment?
Yeah.
So I think that one of our future goals for the bioproduction, especially CNBTI, is some capital production goals. So, I mean, depending on the product that we're interested in, it might well work to solve the cells of nitrogen, they're coming up for a long time, and for other things as well. And it cannot just be programmed for growth into production. It's something that we discussed for 10 years, but it's not an easy thing to uncover.
Kind of like what you were talking about with glycogen storage, like if you could funnel the energy put into storage, not into growth or storage of glycogen, but maybe production of something else. Yeah, that's really interesting to consider.
Okay. Yep.
Awesome. Thank you guys so much. You can totally stop recording.
Okay.
People don't-
Pause recording.